Thank you so much for the nice introduction. Really nice to see everyone. And we have a new baby at home, so I'm not able to travel to visit in person this time, but hope to come back to visit in Ann Arbor in the near future. Um, so I'm really excited to share some directions we're exploring around this idea of building different AI scientist agents, right? And then also exploring how we can try to use these agents to try to accelerate different aspects of biomedical discoveries and analysis. So the context for this is that so AI has been used as a tool for many different aspects of biology and medicine research, right? And it's been very successful over the last 10 years. So for example, if you have like a well-defined problem of like proteins, right? So then AlphaFold is a very useful tool for that. Or if you have a problem of like, let's say, analyzing different microscopy images, then there are different deep learning models based on computer vision that people have developed for doing those kind of cell segmentations and so on. And typically, the way it's used how we use AI is that we first define like a specific problem, right? And then you can develop an AI model to tackle that problem. What's kind of interesting is that over the past year and a half, is that conceptual shift where people are starting to think about AI not only as a tool for solving a well-defined specific problem, but really as almost like a co-scientist that can try to help researchers across a broader range of research endeavors, right? So everything from using AI to help to come up with questions, hypotheses, to designing experiments, analyzing data, and writing papers, and so on. And I think that's quite an interesting shift, right? And a lot of that shift is really driven by advances in we call these AI agents, which are essentially systems that you can build on top of large language models like ChatGPT that then enables these language models to use different tools like AlphaFold, but also enables these language models then to communicate with other AI systems and also with human researchers more effectively. Right. So these kind of AI agents then can be more flexible and help researchers across this broader kind of research and endeavors. I think we're still in the early stages of these AI agents. But I think there are a lot of sort of exciting developments and potential applications. And what I want to do today is just share with you some of the examples of these AI scientist agents that we've been building recently, and also explore some of their applications and also their limitations. All right, so I would love for this to be very interactive. So if people have questions, you can just feel free to interrupt me, and I'll also pause at a few places. So I'll choose 3 examples of these AI example that we'll discuss is what we call the virtual lab, which is a platform where we have a team of AI scientist agents that in many ways sort of mirrors my physical lab at Stanford. So in the virtual lab, there's an AI professor agent, and working with the AI professor agent, there's actually a bunch of different AI student agents with pretty diverse expertise, right, in data science, machine learning, and immunology. And they actually run their own group meetings, and we give them a budget so they can actually do some of their physical experiments. We also where the AI scientist agents can actually go to the school and then teach themselves how to become a better researcher. So I'll explain a little bit more how this virtual lab works. So first, the way that we interact with these AI agents in the virtual lab is that we mostly can talk to the AI PI agent. So for example, I can explain to the AI PI agent some description of the problem that we're interested in. Like maybe I want to analyze a complex genomic data set, or I want to a new protein, right? And then for each of those problems, the PI agent would say, okay, given that problem, what are the areas of expertise that are needed to tackle that task? Right? Maybe for a specific problem, the PI agent might say it's useful to have a computational biology agent or an immunologist agent, right? And then it's actually the job of the PI agent to create and to train the sub-agent that have that relevant expertise. So for different projects, the PI agent can actually have the flexibility then to create and train different agents with different types of expertise file setup. I'll come back to in 2 slides to explain how that training process of the agents happens. But for now, you can think of each agent as basically being like a frontier language model. So think GPT-4 or GPT-5. And then we can do additional fine-tuning on top of these models to teach the models how to use specific tools and more up-to-date knowledge. So after these agents are trained, then are created by the PI agent, then they can start to have their own group meetings, right? So similar to our team meetings, right? Human meetings, agents, they also have their own team meeting where all the agents will meet together to discuss and formulate a research plan. You can also have one-on-one meetings where one of the agent will actually meet with the PI agent and to look at a review of subtask, maybe analyzing a piece of the code or particular data sets. So to make it more concrete, I want to show you an example of what this virtual lab team meeting will look like. So this is actually the first project that we assigned to the virtual lab, which is to help us to develop COVID variants. Right. So at the time of this project, there were some new COVID variants, and we didn't have good binders. So we thought this was kind of a timely and also a challenging open-ended research project that the agents can help. So basically what happens here is that in the beginning of the meeting, so the PI agent then sort of explains to the other agent, like, what is the goal of this project? And after that, the different agents then can speak up one after another to contribute their ideas towards that overall goal. In this case, you saw that the immunologist agents actually spoke up second, right? After the PI agent, and then the immunologist agents actually recommended designing these binders in the form of nanobodies, which is actually a little bit unorthodox already, because most of the time, if we ask human colleagues, they might say let's design antibody binders, which are more commonly used. So nanobodies are sort of small cousins of antibodies, and they're much less common. They're only found in a few animals like camels. But there turns out to be a good reason for this unorthodox idea. So the machine learning agent spoke up next after the immunologist and says that I like this idea of nanobodies instead of antibodies, because these nanobodies are smaller. That actually makes my job as the machine learning agent easier to computationally predict their structures. So you can see how they can build off ideas off each other. In all of these meetings, we also have a scientific critic agent whose job is to basically provide more skeptical feedback to the ideas of these other agents. So here, the critic agent says that there's not a lot of public data sets for nanobodies, right? So then you need to be very careful about overfitting models if you're going to start to design nanobodies. So the actual meeting goes on for longer. Here, I'm just showing the first part of the meeting, but you can already see how the agents are able to respond to each other's ideas and then interact and then relatively quickly converge to like a research plan. Yes, yes, Yichun?
Yeah. Hi, nice to meet you. Glad to have you here. So I got a quick question. So, did you talk about what is the right order of all these agents talk in the team meeting?
Yeah, yeah. So it's a good question. This is where, so the order of which the agents speak does matter. And one thing we can do is that we can actually run multiple meetings in parallel so that the different agents actually speak in different orders. Right. And this is possible because the meetings for these AI scientist agents is actually much more efficient than our human meetings. So one of their meetings maybe takes a few seconds. And because they don't get tired, so we can actually run multiple meetings in parallel. So basically what we do is that for every question that they want to discuss, they discuss it 5 times in parallel. And then in each of the parallel meetings, then they can often come up with different ideas. So then we ask the PI agent to read the transcripts from all these parallel meetings and then come to a consensus of what to do next. So it's kind of a way to explore a broader range of research ideas.
Okay, thanks.
Yeah, good question. Yeah, Andrea?
Hi, thank you. So I really like the skeptical, I guess, agent or scientist there, but I was wondering what other checks for hallucination or just like if they keep, if there's like incorrect data and it just keeps building on incorrect information.
Yeah, so it's a good question. So having the critic agent is quite useful in reducing some hallucinations. I think actually having this parallel meeting structure also helps and look at the consensus across these parallel meetings, and the consensus is often more robust than any of the individual meetings. The third thing that we do is then also to have really the agents do a lot of their work in code, in writing algorithms, and then we can basically go over and then try to debug the code. And I actually have the critic agent to try to run the code and make sure it's robust. So when we force the agent to basically do their work in code rather than just through human language, it's also an effective way to.
Thank you.
Yes, Willis?
Hello.
Hi.
Oh, okay. Thank you for the opportunity. So, how do you make this happen? Like, do you first initiate the PI to make a command, and then you take that and communicate it to all other agents and wait for these responses? Like, what is running all this structure?
Yeah. So we created our own infrastructure, which we open sourced as part of the virtual lab paper. So one part of the infrastructure is basically like this meeting structure so that all the agents can participate in this meeting, and then they can see what other agents say and talk about in these meetings.
Thank you.
Yeah. And this is also related to this following, which is basically showing how often the different participants would speak in these virtual lab meetings. So for example, the human researchers, which like myself, my students, we don't talk very much, so maybe about 1% of the time. So our role is to mostly provide some high-level feedback to the agents. Like we tell them, this is important to design these nanobodies for COVID, or we also tell the agents some constraints that we have, like how much budget we have to do the wet lab experiments. But otherwise, we mostly leave the agents, give them the flexibility to explore their own algorithms. So for example, the machine learning agent in this case, that's all the coding for the other agents, right? So another agent might tell the machine learning agent that implement this new algorithm, and the machine learning agent's job is to actually write that algorithm in Python or R or other languages. And I also, now that we see how the agents interact, I want to also explain a little bit more how the agents are trained through this virtual lab school. So the a school for the agents is that if you take the frontier models like GPT-5, these models are quite good at more broad general knowledge. But if you ask the models to go really deep, right, into a technical domain, sometimes the models do not have the most up-to-date or the deepest expertise in those domains. Right? So they might not know about like the latest clinical developments, right, the latest vaccines and so on. So to mitigate that, right, to fill in that gap, so we created this school where the agents can actually go to the school and then design their own learning curriculums to teach themselves how to become a more useful and better researcher. So the way that first would tell the agents some of the topics that the agents should learn about, right? One topic would be like learn about the latest COVID variants or learn about how to use the AlphaFold tools. And then for each of those topics, then the agents can actually design a set of questions that are relevant for that topic. And based on the questions, then we enable the agents the ability to basically go out and to use different search engines to find and retrieve relevant information. So for example, the agents can actually go to PubMed Central and access papers that are relevant for each of these topics. And then they would download the papers, right? Read the papers in detail. As the agent is reading each paper, then we also enable the agents to basically do supervised fine-tuning, which then they can use to update their model parameters. So under the hood of each of these agents is a large language model. So it's a transformer neural network. When the agent is actually reading each of these papers, then they can update the weights of the transformer to help them to ingest that new information. So initially, the agents might start off base model, like GPT-4o model, right? But after they go to the school, then they start to diverge, right? So the agent then for computational biology would have a different parameter, right, compared to the agents with expertise in immunology. And it wouldn't be a very good school without some sort of testing mechanism. So after they go to the school, after they go to the training, we also have a teacher agent who designs quizzes to test the students, right? And then if the student does not do very well on that assessment, they have to go back to school readings and fine-tunings before they can pass the quiz and graduate, right? And after they graduate, then they can join the virtual lab and work on these research projects. So one thing is that the agent school is also quite fast compared to human schools, right? So it takes maybe about a couple of days for the agents to go through and finish the school, and then they could graduate and then start to do. Okay, so now that we see how the agents interact and their schools, I want to come back to this example, right, to illustrate the kinds of research that they are able to do. So recall that the first project we give to the virtual lab is to basically help us to design binders to the recent COVID variants. And they decided to design these binders in the form of nanobodies. Now, there's not a lot of public data sets for nanobodies for COVID, right? So to overcome that data, the agents decided to create a new computational workflow to help it to optimize these nanobodies in silico. So here's the workflow, the computational pipeline created by the AI agents. Right, so first, the AI agents decided to use this protein language model called ESM, right, to look for candidates. Right? So ESM would take as input an amino acid sequence and then computes a score to say how likely is that sequence to form a stable functional protein. Right? So this would be the first filter used by the agents, and then the agents would pass pass it into AlphaFold together with the spike protein of the target COVID variant. So AlphaFold generates this three-dimensional structure, and the agents would look at that three-dimensional structure and extract some scores to see how good a fit that is. And then the agents will pass that three-dimensional structure from AlphaFold into Rosetta, which does for like a physics-style relaxation and estimate the binding affinity. Right? Then the agent will also extract from Rosetta like a binding affinity score. So these different tools use methods and have different information that are provided. So the agents actually take the output from each of these tools and then combine that into like a holistic scoring mechanism to decide which variants, which nanobodies to prioritize, right? Then they would iterate through this pipeline multiple rounds to come up with maybe the most promising candidates. All right, so these individual tools have previously been used and published in previous papers, but the agency can also make some modifications to these tools, right? So for example, the agents actually modified the ESM, so it's different from what people have previously published. And then this specific computational workflow created by the agents is also somewhat different from some of the workflows that people have previously used. So this is all in silico work that's sort of designed and implemented by the AI agents. But the agents, they don't do their own wet lab experiments. Right? So this is where there's also an important aspect of human AI collaboration. So the agents basically told us like a list of that they thought was the most promising. And then we, with our experimental collaborators at the Chenzek-Berg BioHub, then we can synthesize each of these 92 nanobodies in the real world and then test them in the wet lab to see how effective they are. So here are two nanobodies that are designed by the virtual lab agent. So these are new nanobodies, so people haven't seen these before, right? And then we actually made these nanobodies, and then we tested them in these ELISA assays to see how well do they actually bind to different COVID. What these experiments look like in the bottom. So if we look at the bottom red curve with the open circles, this is where if we take human design nanobodies, right, and then test those against a recent COVID variant called JN1 variant, right, it does not work very well. The second red curve are the nanobodies designed by the virtual lab agents, right? These are new nanobodies. And you can see that they actually they do show more promising binding to this JN1 variant. And the blue curves basically tested against a very different strain of COVID, right? This top one is the AI designed nanobodies. They actually also show pretty good binding, right, to this Wuhan strain of the virus, which came from 5 years ago, compared to the human designed nanobodies. So this gives us some signal, right, that the promising signal that the virtual lab designed nanobodies shows promising binding across this quite broad and diverse spectrum of the COVID viruses. And we did, it's not shown here, where we test these nanobodies against other viruses and also host human proteins, and they do not bind to these other proteins. So these are binders that are specific within this COVID family, but within the COVID family, that seems to show quite this the broad spectrum binding, which is quite exciting. Now, the caveat here is I think there needs to be a lot more experiments to further validate and characterize these binders. And actually, one thing we've done is to also we've given these back to the AI agents. So they've taken that and then further optimized their computational pipelines. So there's going to be like, there can be this experimental in the loop process.
James?
Yes.
Yeah, Dr. Omenn had a question in the chat. How do you synthesize the nanobodies?
Yeah, so this is, we actually used a CRO to basically a contractor. So we give them the designs from the virtual lab, and then they synthesize the nanobodies for us.
Thanks, makes sense.
Yeah, thanks. Yeah, so the agents currently, they don't do their own wet lab experiments. So they basically have to hire contractors to do those experiments. Cool. Yeah. So just to summarize this first part here, so we released all the open source as a part of our recent paper, and people can now use it to actually tackle quite different kinds of research problems. So they're not limited to designing proteins, right? People are now also using it to, like, for example, look for biomarkers, Alzheimer's, and other applications. And we also found in our experiments that when we have teams of multiple agents with diverse expertise, that often works much better than if you have a single agent to tackle the problem by itself. Basically, one thing will happen, when you have multiple agents and they have meetings together, sometimes they get into disagreements or debates, right? And that often sharpens the reasoning process to make it more robust. Whereas if we just give a single agent a task, then often the agent just try to go through it by itself without too much debate or internal discussions, and that often is less creative or less robust. We also found it to be quite useful to provide the agents with additional resources. Like, for example, we have a memory mechanism that we provide to each, sort of like a digital notebook, so they can remember their previous discussions. Each agent also has like a local sandbox environment, kind of like a Docker container where they can actually locally use different tools like AlphaFold and make modifications to those tools. Right? So this provides kind of like a safe environment where the agents can actually run, build their own tools, do local experiments without the risk of maybe the agents actually contaminating the broader ecosystem. Right? And interesting that the agents have their own social dynamics. So by that I mean, when humans collaborate, right? So it depends on, some people might talk more than others, and some people might have louder voices, and that all changes the trajectory of the research and collaborations. We also see that with the agents. So some agents talk more than other agents, and depends on also who speaks first or second, that also changes the discourse. So one thing that we could do with the AI agents that is hard to do with humans is that we can actually do all sorts of counterfactual. We can change the number of scientists on the team, right? We can also change the personalities of different agents. And then we can also see, we run the experiments in parallel and see, how does that actually lead to different kinds of results and science. And essentially, it's almost like a petri dish where we can try different kinds of scientist agent teams and see what combination of AI scientists actually leads to the most robust and creative solutions. So I'll pause here and see if people have any further questions before I move on.
Hi. I do have one question. This is so interesting, so fascinating. In general, I know that usually like when computationally identify like a new structure, new nanobody, or like new things, potentially like it's like that new thing require new experiment to validate. But sometimes like when it's expensive or hard to get someone to really test on the specific one, people do try to like see whether previously known working molecules, working structures, whether they appear rank high in some of the rankings, or whether they also like can be predicted by the new algorithm. I wonder whether it's like the agents can also, or whether it's like they can also do something like that, like try to find previous literature, whether they're like known working molecules, and maybe also use that to like further improve the algorithm that develop.
Yeah, it's a great question. So here, it's a little bit challenging for the agents because there's not a lot of existing data since it's a new COVID variant, this JN1 variant. And in fact, there's like an existing nanobody designed by the human team, but that one, as you see here in the experiments, actually did not show any binding signal to this recent COVID variant. So this is actually where the agent did look into the literature, and it was pretty challenging. And that actually motivated them to come up with this computational in silico design, where they can actually use the AlphaFold and Rosetta, these other tools, to help them to like in silico optimize these nanobodies, precisely because there's a shortage of public data, experimental data.
Okay, thank you.
Yes, Todd, I think.
Yes, James, great to see you again. This is really impressive work. I was wondering if you had tried giving the model slightly more open-ended questions where it actually is trying to identify an area of research that may be, fertile ground for innovation and scientific discovery, rather than giving it a very targeted question.
Yeah, it's a great question. This is actually maybe a little bit more like what I'll show in the second example, in the second part, where we have the CellVoyager agent whose job is actually sort of look for new questions beyond what people have previously asked.
Sounds good.
Yeah, I'll come to that shortly. Yeah, thanks.
Hey, James, we have a question. So for training these agents, one, what do you do for quality control of the things they learn from? And two, is there a memory limit, or would you suggest a memory limit to what you feed them to learn from?
Yeah, so both are good questions. So with the quality control, we mostly rely on the quality of different search engines. Okay, so for example, the agent will actually go to like PubMed Central to look for articles and then basically find like the top articles returned by PubMed. And there's going to be a bunch of noise in those articles, in the literature itself. This is where we mostly rely on the agent themselves to figure out how to like reconcile maybe conflicting information. But ultimately, the agents are evaluated based on their performance on the experimental ground truth. And for the question related to the memory, so yes, so we have an external memory mechanism for the agents, right? So that means that they can then remember more things than they could just keep in their own model. So that's enables the agents to, for example, remember discussions from all these parallel meetings, and also from their previous one-on-one meetings with the PI agents.
There's another question.
Okay.
Hi.
I have a follow-up question about the memory mechanism. Like basically you mentioned relies on the meeting summaries or carrying the context across discussion, but doesn't seem to have a persistent memory of the prior experiments or the previous failures. Do you think incorporating a long-term memory mechanism would make the system more autonomous across multiple projects or like previous like exam
Yes.
failure case, and is that more promising future direction?
Yeah, I think these kind of lifelong learning type techniques will be very useful for AI agents. Like in this case, for example, now we have some experimental results. We actually give those back to the agents. So now they do have memories of these, and then they can use that to further refine their computational pipeline. But similar to humans, like the more experiments they do, success and failures, then we also wanted the agents to learn from all these other projects. So I think this kind of lifelong learning memory would be very useful. Great.
James, I have a question that is sort of or not very related to this, because this is kind of a community, like a committee-based decision using AI is quite cool. Oftentimes we complain about the, in the HPR reviews, very flawed and biased.
Yeah.
And I was wondering, what if we have, AIs that are trained as different experts, just like immunologists and such, and critics, and then put them together and see what kind of results will generate as compared to human experts review scores. That would be a very interesting thing to see.
Yeah, I think that'll be really interesting. Having like a agent, especially with the government shut down, right? So maybe it's hard to find the human review panels now, but I think that would be a very useful application to have the agents try to do those kind of reviews.
Yeah, another related question is, since you mentioned about the social dynamics among the agents, so I was wondering, that the critique person, that critique agent, how does that critique role helps guiding the direction of the antibody selection at the end?
Yeah, the critic agent we found to be actually really important. We did some experiments where we removed the critic agent, and then the other researchers start to make a lot more mistakes, right? So the critic agent is actually really important. For example, we often have the critic agent to meet with the machine learning coding agent to review the code that they write, and the critic agent also finds like a lot of bugs in the code, right? So then they can fix it together. So the critic agent, I think, is actually a really critical, important piece of the virtual lab.
Thank you.
Great. Yeah, thanks for the great questions. So then I want to, in the second part, talk about a related and recent project we did where we tried to build specifically actually AI data science. And the goal of this agent is to really try to help us to maybe explore unexplored areas in data science, right? And also come up with new insights. And we want this agent to be take its inputs, quite complex data sets, which could be like, for example, transcriptomics, single cell data, and the agents actually then generate these detailed notebooks, Jupyter notebooks. So each notebook corresponds to like an in-depth analysis of the data set. And then the results from these different notebooks into like a report with reproducible figures and interpretations to describe what are the new insights that they've learned. So the motivation for why we think this is a good use case, this kind of data science is a good use case for agents, is that first, there's this increasing amount of really high throughput, very complex data. right? And in parallel, there are also this really rapidly evolving ecosystem. Right, so just for example, in single cell analysis, where we do a lot of our work, there are also already like thousands of different potential tools that people could use. And these two things combined often makes it quite challenging for the practitioners and users, right, to say, okay, so what tools and what analysis should I do to make sense of my data, right? As a result, often people might, they spend a lot of resources collecting a very complex and rich data set, but they might only be able to scratch the surface of what can be contained in the data. There's many important, interesting insights in the data. So we hoped that so this motivated us to try to create this AI agent that can help the researchers to try to explore data sets that they have already collected, perhaps in new ways that they may not have thought of before. And as a result of that, maybe hope to generate new insights. Right. So the input into this agent then would be the data set itself, and the output will be a new report with figures and also reproducible code. So the way that this CellVoyager agent works is that first it takes the input data sets, right? It actually does some exploratory data analysis to understand what is in the data and what's the metadata. And then it does a deep research to understand how have people in the field have analyzed similar data sets before. And based on that, it can prepare like a Jupyter environment by importing and initializing the relevant tools. And after that preparation, the agents come up with like a list of hypotheses, right? And these hypotheses are designed to be quite different from each other, right? And for each hypothesis, an agent will then execute like a set of step-by-step analysis, right? Where it would generate some code to analyze the data, run that code to generate the figures, and then actually uses like a vision language model to interpret those figures and decide on what to do next, right? So each analysis then can go quite in depth into some particular aspects of the data. So here's an example of this analysis done by the CellVoyager agent. Right? The agent does, it's in the form of the Jupyter notebook, and that makes the analysis easy to reproduce and also more transparent to the users because you can just go back in and modify the notebook and rerun the different codes. So, and the notebook produced by an agent is also relatively structured, right? So it always starts with some description of what is the hypothesis or the idea of the analysis, right? And then the agent will implement the code based to implement that hypothesis, and then block right on the data to generate some figures, and then it will interpret those figures to see what the insights are learned from this analysis and from the figures. And based on that, you can iterate and decide the next analysis and next code and so on. So this enables the agent then to iteratively analyze the data sets in greater and greater depth, and it will do this in parallel across multiple different analysis threads. So the way that we the CellVoyager agent is by essentially asking it to, by giving it some papers that people have previously published, and also with corresponding data sets from those papers. These are pretty complex data sets. And then we ask the agent to basically reanalyze these data sets, but come up with new insights that were not in the original papers. And to validate this, then we give the report from the agent back to the original authors and have the authors themselves to help to validate for us that it's done correctly. And then second, the analysis from the agent are indeed in new directions that they themselves had not thought about in their initial publications and initial analysis of the data. So here's an example. So we did several case studies, but here's just one example illustrative case study. So here we have a paper, a previous publication, which is a nice study on brain aging. So it basically sequenced the single-cell RNA sequencing of brain cells across different ages. And the goal of the original study is to basically build aging clocks, right, based on the single-cell transcriptome. And here CellVoyager performed 5 new analyses. And we give these back to the authors, and they thought 05 was done correctly. And then in particular, 4 of these 5 analyses by the agent was actually quite new and interesting from the perspective of these original authors. So here's one example of this new analysis. Figures generated by the CellVoyager agent. So basically, typically the agent would do some data exploration first, right? So here it's dealing with single-cell RNA sequencing data. So the agent will do some visualizations and clusterings. And then it started to dive pretty deeply into some interesting new hypotheses, right? So here the agents actually found a pretty interesting and consistent correlation between transcriptional noise and biological age. So essentially, the agent found that cell types, right, cells that are older tends to have higher transcriptional entropy. And this is actually a pretty interesting finding. This correlation between transcriptional entropy and biological age has been observed in other tissues and organs, but not well studied in this part of the brain, right? And here the data actually came from the brain. So this is sort of a setting where, and the original authors of the paper actually did not know very much about the literature of transcription, didn't think of doing this analysis. But here, the agent, by reading the relevant literature, is able to do this new analysis, actually find kind of this interesting pattern. And then the agent is also able to find like a second paper, a different paper and different data sets. And they also analyze the second data set, which is also single cell data from the brain, right, to further validate this finding in sort of an external cohort, right, which makes this like more robust from a data science perspective. So we thought this is kind of an interesting example of like an agent essentially reanalyzing a previously published data set, but finding a new insight that was missed by the original authors. But the agent is then able to further validate based on literature and also based on external cohorts analysis. Great. Any questions about this second part before I move on? Yes, Ichen?
Oh, yeah. So one quick question is like, oh, let me turn my video on. Right. So quick question is about, so is this finding the only finding in this process, or this finding is among many other findings, and how did we filter this finding out if that's the case? Thanks.
Yeah, so thanks. Yeah, so here the agent actually did 5 findings. So did 5 analysis in parallel. And then what I did 1 of these 5 analysis. So when we have the human authors review this, they actually found like basically 4 out of the 5 analysis to be novel and interesting from their perspective. Okay, Ulas?
Hi again. So assuming I want to perform a similar analysis on another paper, how feasible is that for me as someone who is not a computational scientist, like average PI in a field, to use the tool you provided to perform this? And what will be the cost?
Yeah, so it's pretty easy now because with CellVoyager, it's also open source, so you can give it a new data set and then have it analyze it. You can give it instructions just to human natural language, so it doesn't require coding. And then it's like maybe a couple dollars, usually less than $2 per like data set analysis.
Thank you.
So that's actually a good question that sort of motivates also maybe the third part, the last part I want to mention, which is a quite recent work that we just released that we call Paper to Agent. And the goal here is to try to reimagine what like knowledge dissemination would look like, right? In a traditional standard way, right? So people would do a research and then write a paper, right? Maybe you have a big PDF. And the paper, if you think about it, is, even though it's a standard way for capturing knowledge, but it's really very passive artifact of knowledge, right? If you have a paper, then it's actually a lot of the work for the readers to figure out and actually understand your paper. And if your paper releases data, it's often quite a lot of work for the users and readers to figure out how to use the code or how to use the data, right? So there's a lot of technical barriers. And our idea is that instead of having these sort of passive artifacts of papers, why don't we try to turn each paper into an interactive agent so that the agent then becomes like an expert, so knows everything about the paper. If the paper has some code, then the agent is sort of the expert in using that code. So if the reader wants to apply some methods or data from the problems, they can just ask the agent to help them to do that, right? So in this case, right, you can just ask the paper agent, can you apply your method, right, to a new problem, right? And then the agent would just interactively do that, right? And generates sort of reproducible analysis for the user, right? And provides some interpretation of the results, right? So this will all be done interactively without the user needing to download and try to set up the environment on their end, the code from the original paper. So you can basically view this paper agent as essentially like the virtual corresponding author of the paper that's available 24/7 to help any of the readers to apply the methods and the data from that paper to their own problems. So the way that we do this, right, is by the way that we convert papers to interactive agents is that we have this automated workflow that we created for the paper to agent workflow. So briefly, the way that this works is that we essentially have worker agents. So for each paper, these different worker agents will actually go into the paper and into any of the code base, like GitHub pages released as part of the paper in depth. So one of the worker agents' job is actually to set up like the virtual environment. And another worker agent's job is to basically extract all the different tools from the code base and from the paper. Then a third worker would try to create a bunch of different unit tests to make sure that the virtual environment is robustly implementing in the paper itself, right? So this will all be done automatically. And the output of this is what we call a MCP, or a model context protocol. So recently, these MCPs have emerged as sort of like the industry standard for providing additional external resources to communicate with large language models. So here, essentially, what we converted the original paper into a paper MCP, right, in this automated workflow. And then this MCP then contains basically all of the tools and insights from the paper, right? And then we can connect this MCP with like a downstream chat agent, or could be your favorite, like ChatGPT or OpenAI or Anthropic models. And by connecting this chat agent, right, with the paper MCP, this essentially enables the chatbot to be an expert in using everything from the paper and the corresponding code and data. Right, so this is sort of paper to agent workflow. So just a couple of quick case studies, right? So the ScanPy is a quite used package for all sorts of single cell analysis. So with paper to agent, we can then automatically create the ScanPy agent, right? So then the ScanPy agent then will actually knows about all the different functionalities of ScanPy, right? So like a user, instead of having to run ScanPy on their own laptop, right? They can just ask the agent, or can you apply, do this kind of standard single cell analysis on my own new data, right? And the agent would actually correctly use ScanPy, right, to do the. Right, so here on the left, basically we show the ScanPy agents produce analysis results, right? And on the right would be analysis results from human expert users, right? And they, in this case, they match exactly, so the agent's able to essentially reproduce analysis from human users, right? But the usage of this ScanPy agent is not necessarily to help the human experts, but really help those other biology users who experts themselves, right? So then those users, non-technical users, then can use the ScanPy agent to help them to do their own data analysis. We can also use this alpha genome, this paper to agent to basically convert very recent works, right, into agents. So alpha genome is quite a recent foundation model put out by Google DeepMind. It's quite a powerful model that can basically help to interpret effects of different mutations. With the paper to agent, then we can also create automatically an alpha genome agent. And then you can very easily use the alpha genome agent. So for example, here you can just ask the alpha genome agent to, can you try to find causal genes related to this variant, such as LDL cholesterol. And then the agent, because it's an expert at using alpha genome, right, it would take this query from the user, right, generate sort of an analysis plan, and then iteratively use alpha genome, all these intermediate results, and finally provides like a result back to the user, which says that, okay, sort 1 seems to be the most likely causal gene near this variant. Right? So all of the steps will also be transparent and reproducible for the users. They can go back in and check or rerun each of these steps. But we think of this agent now as really much easier to work with than if the reader has to just directly read the alpha genome. It's a 100-page paper, try to figure out what to do itself. So I'm quite excited about this idea of the paper to agent, really as a way to try to reimagine what knowledge dissemination, what publication should look like, right? So instead of having just purely passive paper artifacts, right? What if we convert each paper, right, also into this interactive agent, and then the agent then can help all the readers and users on how to use the insights and the methods and data from that paper to new problems. So this can make the knowledge much easier to be disseminated and also make collaboration much easier. And you can also imagine other ways of using the paper to agent, right? Maybe you can also use it to help with reproducibility, right? So maybe if the paper does some analysis, you can have the paper agent try to see, can you reproduce this analysis before it's officially submitted for publication, right? Great. And I mentioned so putting these different pieces together. So we're currently also running this kind of interesting experimental conference called Agents for Science, where for the first time, both the authors and reviewers are AI agents. So the motivation for this is that the existing conference journals and conferences do not allow AI as co-authors. So we thought we will flip this around so that in this conference, you have to be an AI to submit. Humans can be co-authors, but AI needs to be the first author to do most of the work. And then also by multiple other AI agents, we'll have human experts do the final assessments. So we've received over 300 submissions from different agents to the conference. And because the reviews is also done by AI, so the reviews are finished within a day. So now we're doing the sort of the final human assessments for the top papers. The conference itself will be in a couple of weeks in later October. And will also, everything will be freely and openly streamed. These, like all the papers and reviews, a sort of open resource for the broader community so that everyone can then see how good is AI at doing research, like what are the weaknesses or the strengths, what are the mistakes the agents make, and also how good are the AI at reviewing and catching those mistakes. And the last part I want to mention related to that is that I think it's also important to think about what are the limitations of the current AI agents and how that complements human expertise. One of the current agents is that I think the agents are better at using tools that people have already developed rather than creating entirely new tools from scratch. Right? For example, AI agents can learn to use AlphaFold quite effectively, but it's going to be much harder for the AI to create the new version of AlphaFold by itself. So this is, I think, quite complementary to a lot of the human researchers and expertise, right? So a lot of the people here in this audience are developing a lot of exciting new methods. How to get those methods to the right users, right? And help the users use those methods. So this is where I think the agents, for example, things like paper to agent can help to make these methods that we develop much more accessible, right? So I view the agent sort of the interface between the new methods that human experts develop and then a lot of the downstream application users. Similarly, the current agents, I think, are more about breadth rather than depth, right? They know a lot of different areas, but not really very in depth for a particular thing. I think this is also quite complementary in that a lot of the human experts, we go very deep into a particular domain, try to be very creative in that technical area, but sometimes we don't have the breadth of all the adjacent areas. So this is where I think there's also quite complementary strength and weakness where the AI-human collaboration can be quite useful. The agents, they make mistakes. Often we found that the mistakes happen because the agents don't really fully understand the context of the problem. So for example, if we give they analyze, and if they don't tell the agents how the indexing values in that data set are imputed, right, then the agents can make assumptions that are not valid. So this is also where it's quite critical for us to try to really capture a lot of the metadata in kind of a form that's easy for the AI to ingest, and also to provide critical assessments of different critical important steps of analysis from the agents. And of course, I think because the agents are mostly limited to in silico analysis, even more, I think, important to have good experimental collaborations and experimental validations. Cool, great. So I'll wrap up here. Just to summarize, I hope to show these 3 examples to illustrate while we're in the still early stages of the AI scientist agents. I think there are a lot of exciting potentials for these agents to help us to accelerate discovery. Right. So in the virtual lab, we've seen how teams of different AI scientist agents can and tackle quite challenging problems like designing new nanobodies, which we have experimentally validated. In CellVoyager, we have these computational biology agents that can really help us to do more systematic data analysis, and they're able to find new insights that we have missed ourselves. And with papered agents, we're sort of trying to reimagine what publication and also knowledge would look like by converting these sort of passive paper artifacts into more interactive agents to help with collaboration. So I want to especially acknowledge my students that led each of these different projects. So Kyle Swanson for Virtual Lab, Sam Elber for CellVoyager, and Jashin Mel for Paper to Agent. Thank you very much. Happy to take any further questions.
Wonderful. Okay, somebody has already raised their hand. Okay, go ahead.
Yeah, really nice talk. So I got a question. So with so many agents, what are the issues? What are the issues do you think in the system level? It's like costs or performance, and also, do you think that's the important issue?
Yeah. So it's a good question. I think a lot of these agents can actually be relatively cost effective. So for example, for the virtual lab experiments, the computational part of it is only like a few $100s, right? So the experimental part, like actually making the nanobodies, ends up being like orders of magnitude more expensive, right? So the agents can actually be, I think, relatively effective here. I think there will be some new emerging systems questions. Now imagine now if like every paper becomes an agent, right? So then there'll be many thousands and thousands of different agents, right? And how do we think about organizing that community, that ecosystem of agents, right? So it's efficient for us to like search across the agents, and maybe how do we also facilitate agent-to-agent communication? I think those are some quite interesting emerging new also research topics.
Thanks.
Yes, Zhao Wei.
Yeah, hello, very fantastic work. And I'm very interested in like paper to agent. So for example, if we have like a random paper, I'm actually very interested in like what is the successful rate for your like automated workflow that can translate this as an agent?
Yeah. So if the paper itself, or if the code base of the original paper is not very detailed or very well documented, then then paper agent will have a hard time converting that into a robust agent. So in that sense, we view that this like paper agent itself is almost like a metric to see like how well documented or how reproducible is the original paper. Like if the original code base is missing so much details that it cannot be identified, then that itself may be like a sort of flag that maybe there's some other issues and perhaps like there needs to be more documentations in the original paper.
Oh, sounds good. Okay, I have another question. Like for CellVoyager and also paper to agent, I feel like there's a very long procedure in planning. So I wonder like how could you like solve some error accumulation, or sometimes like ARM agents will stuck in a loop that cannot solve, or encoding bugs. So I wonder, is there any mechanism like you solve this error accumulation?
Yeah, it's a good question. So this is where we also find some benefits in having a multi-agent. So here, for example, with papered agent, we actually have multiple different workers, right? Each of the worker is having a different specific task, right? But they can be run in parallel and complementary. Right? For example, one of the workers' tasks is specifically like a testing agent, right? So essentially tests like how good is the MCP that have been produced by the other workers, and then that can be used then as a way to optimally like, further refine and robustify the MCP.
Okay. Yeah, I have another question. Actually, I'm also working on this field. So I wonder, do you think like agentic RL will help like to solve this error accumulation, or does it have like a promising future? Or are you considering like using these kind of techniques?
I think there are definitely a lot of promising applications of RL for agents, right? But ultimately, like it's one of the challenges RL itself is this credit assignment problem, especially if you have a very long planning path, right? I think this also be challenging to figure out which part of the path, right, should receive different kinds of credits. And that becomes even more challenging when you have like multi-agent RL workflows, right? So I think there still needs to be improvements on RL side to really help with this very long planning.
Okay, thank you so much.
Yeah, so related to this, I was thinking about the reverse problem. You actually touched a little bit. If you have agents coming out with learned knowledge from individual papers, and then how would it be harnessing the knowledge is learned from different agents and then try to synthesize new knowledge for research purpose, for example?
Yeah, it's a good question. Like I think, like for example, with the paper to agent, right? Let's say if somebody publishes like a new methods paper, then we turn methods agent, and somebody else in a different country publishes like a new data set paper, and then we can also convert that data set to like a new data agent, right? And then the two agents can actually just directly communicate. That's what I would like to see, right? The two agents, like the methods agent can then just talk to the data agent and form their own collaboration and then apply the new method to that new data set without the humans having to each other, right? Because that's often the sort of bottleneck for us right now, right? So I'm excited about this sort of world where all of this different research that we produce will be agentified, so they become very interactive. And then the different pieces of research that you do or I do, they can just directly talk to each other and collaborate without, we have to try to schedule all these meetings, and because our bandwidth are much more limited.
Right, right. Perhaps on top of that, also add openness versus security, agent in there to governing the data flow as well.
Yeah, that could be very useful. Yeah.
There are many people. I'm kind of lost who raised their hands first.
I think maybe Honey.
Thank you so much for your great talk. How do you quantitatively measure the accuracy of virtual lab or AI agent? For example, when the agent provides information, how do you evaluate how reliable it is?
Yeah. So it's also a good question. So we use different evaluations for each of these different agents. So for the virtual lab, the ultimate evaluation comes from the final wet lab experiments, right? Like the agents proposed 92 nanobodies, each of these 92 nanobodies, and we found that maybe there's a few of these nanobodies that ends up being somewhat promising, and there are two that's the most promising when we do the wet lab experiments. Right. With this paper to agent, then the main way we evaluate would be like, is it able to like reproduce the same analysis, right, systematically that human experts are able to generate, right? So then we can just compare the figures and compare like the statistics and see do they match.
Thank you.
There's Andrea.
Hi, thank you for your talk. So I have two main questions, but I know about time. Both are more high level thinking, both globally and academic. Like I was wondering how your vision, I guess, looking forward for restructuring and academia and higher level education impacts with this virtual lab. Like I'm thinking maybe a program would be looking for like virtual, like training on virtual lab designers, more so like training the scientists, one. And then second, globally, a lot of research comes from different countries and languages. So I was interested in like virtual lab languages, right? Where we can collaborate with among different languages and stuff like that.
Yeah, so both are good questions and certainly important topics. So for the virtual lab side, we really believe that this should be a human AI collaboration, right? That researchers should be in the loop. So our vision with the virtual lab is that imagine like every PhD student or every researcher has like a virtual lab agents behind them with different analysis, different experiments, right? So this could actually potentially make the researcher more more productive, but also help them to focus maybe on the more interesting tasks, right? And ultimately, I think the question related to language is also quite important, right? I think the current models, they're quite good at more common languages like English, Spanish, Chinese. I think it's quite good. We have seen that for less like the ability of these models can sometimes suffer. so that's still like an area for exploration.
Great, thank you so much.
Thanks. Maybe we
Xinhai,
could, yeah, Xinhai.
another question from you?
Yeah, thank you for the fascinating talk. You mentioned that AI agents are mostly limited to like the in silico work, where the human control more like experimental validation and creativity. So looking forward, how should we like evaluate the effectiveness of these human AI teams? And also related to that, I think the AI scientist conference you organize is quite I don't know how to describe. It's very nothing like in parallel, like really. So do you see this is actually a meaningful way to benchmarking or evaluate the contribution of this human AI collaboration in science?
Yeah, I think so. That's actually one of our motivations for this conference. So at these conference, we also ask each paper submission to have a detailed description of how the AI work together with the human, right, in every aspect of research, from hypothesis generation to like analysis and writing, so on. So I think that itself will be a very useful data set to see how do the different human researchers collaborate with AI agents, and then we can directly correlate that with the quality of the research, right? So I think there's a lot of things that we can learn from that exercise.
Anthony?
Hi. So one thing I was noting throughout your presentation as part of the different roles, you relied on either very well-structured data or well-documented data. Have you had tested with an ontologist as part of an agent or some other form of ontology as a way to assess these ideas? Or have you just been truly using the LLM agents, run the code, and then see if it works, yes or no, to spit out the results?
Yeah, so I think it's interesting to look at different ontologies. So here we haven't looked into that so much. We have the agents look at different kinds of data, different modalities, right? So for the virtual lab part, this is mostly protein data. So CellVoyager is a lot of like transcription expression data, right? And then the singles, like the paper to agent, there we applied it to more diverse kinds of papers, including like GWAS papers, so across many different kinds of biology. Right? So I think these kind of agents can be pretty broad across different types of data types, but we need to do more tests.
Okay.
Great.
Thank you.
Great. Well, thank you very much. I don't want to take up too much more time for people. Thank you very much. Great questions.
Thank you so much, James. It was really wonderful to see even more progress, more advanced, and really cutting edge from your lab. Let's thank him for his time and, hope to see you soon in person.
Yes, I love that.
Thank you. It's an awesome talk.